I'm very pleased to participate again in an omics conference. These have been valuable exercises, bringing together people from a variety of disciplines, but which have people who have specific interests and coming together from a variety of specialties. I'm going to talk about thyroid hormone as usual, and here we're talking about thyroid hormone as a distinctive disadvantage in the setting of cancer. I think we all know that estrogen in the setting of a patient whose tumor expresses estrogen receptor, or the patient, the male patient who's still producing androgen who has an androgen receptor expressed by a tumor, are at a disadvantage. What I'm going to emphasize again today is that the thyroid hormone is a wonderful advantage when we're healthy. But in fact, its pro-angiogenic and anti-apoptotic actions are a disadvantage in the setting of cancer. I'm going to need my glasses as well. Thyroid hormone is obviously a non-peptide hormone. It's a diphenyl ether. This is a very uninteresting depiction of the compound. Actually, it is structured like this. The iodines in thyroid hormone prevent the outer ring from rotating around the ether bond. The alanine side chain is essential to its activities in the nucleus. It is not essential to its activity on the cell surface. T4 is viewed as a prohormone, that's at the top of this slide, as a prohormone for T3 in the nuclear actions, but in the extranuclear actions or non-genomic actions, T4 is in fact a hormone, not a prohormone. Tetrac, shown to you at the base of this slide, you'll see is missing a carbon and an amine from the alanine side chain. It is an antithyroid hormone on the cell surface. It is a prothyroid hormone inside the cell. So we're talking about different lives here of three thyroid hormone molecules. As I said, acting inside the cell, thyroid hormone is a critical metabolic and developmental regulator. Acting at the plasma membrane receptor that we described a few years ago, it's a proliferative factor for tumor cells and for dividing endothelial cells. The hormone has anti-apoptotic properties that I'll show to you, and these begin at the same cell surface receptor at which the pro-angiogenic actions occur. Integrin alpha V beta 3 is one of 24 integrins. It's expressed primarily by dividing cells and thus concentrated and activated in tumors. It's usually seen as a sensing mechanism for extracellular matrix proteins. Vitronectin, fibronectin, osteopontin, von Willebrand factor all bind to integrin alpha V beta 3 and are interpreted by alpha V beta 3 for the cell. That is, they're recognized and create specific signals on the cell surface that cause the cells to behave in certain ways when these extracellular matrix proteins are recognized. Now, it's also been recently appreciated that small molecules bind to the integrin, and these include resveratrol, dihydrotestosterone, and thyroid hormone. Here's a cartoon from the Bruce Alberts text of an integrin that's activated. What you see is that it has feet inside the cell that generate a variety of kinase-dependent signals inside the cell, and you'll see lobster claws at the head of this molecule and the legs called the thighs and calves of this compound extending down to the plasma membrane. We've identified the location of the thyroid hormone receptor, shown here in red at the base of the lobster claw head of this molecule, this heterodimer. It's a complex receptor, the thyroid hormone receptor on alpha V beta 3. It has an S1 domain. We've designated this as an S1 domain that binds T3 exclusively. And this is an interesting site which has very specific actions inside the cell. There is an S2 domain that binds T4 primarily and T3 secondarily. It has a very high affinity for T4. And it's the second domain, it's S2, that is in fact critical to cell proliferation. Here are some of the actions of thyroid hormone that begin, biochemical actions that begin at the cell surface. Receptor on alpha V beta 3. You can see that MAP kinase is turned on, that PI3 kinase is turned on. A host of protein trafficking effects occur from the cell surface. For example, thyroid hormone drives the estrogen receptor into the nucleus. It drives thyroid hormone receptors into the nucleus from cytoplasm. It drives STAT3 and other critically important coactivators into the nucleus. If we summarize these effects of thyroid hormone, I've shown this in a larger cartoon, but they're summarized in the left lower corner of this slide. Thyroid hormone acting at the cell surface controls protein trafficking, and this is T4, not T3. T4 controls protein trafficking. It controls serine phosphorylation and acetylation of nuclear proteins. This includes the estrogen receptor and the thyroid hormone receptor. It causes specific gene transcription. More than 30 genes are regulated from the cell surface, almost robotically, by thyroid hormone. And it alters the sensitivity of ion transporters. This includes the sodium proton exchanger, NHE1. It includes sodium potassium ATPase, and it includes calcium ATPase. Now, the complex anti-apoptotic actions of the hormone are desirable when we're healthy, and neurons and myocardiocytes are protected by this anti-apoptotic effect of thyroid hormone. In the setting of tumor cells, however, we would question the utility of this, except to the cancer cell. No utility clinically. Evidence in support of the anti-apoptotic activity of thyroid hormone came first from some studies that we did of an apoptotic model where we used resveratrol to induce p53-dependent apoptosis. This requires serine 15 phosphorylation of p53. And it's an interesting model in that it includes the induction of intranuclear COX-2, cyclooxygenase 2, a pool that's quite distinct from the constitutively expressed form of COX-2 that's a marker of aggressiveness. This pool of COX-2 is entirely different and is in fact a part of the apoptotic process. And a problem here is that thyroid hormone blocks this effect of resveratrol. You can't induce resveratrol. You can't use resveratrol to induce p53-dependent apoptosis in the presence of physiologic concentrations of thyroid hormone. Some of you may recall that GlaxoSmithKline invested heavily in resveratrol as an anti-cancer agent, and in fact the agent failed to work well in the clinical setting. And one of the reasons we believe that resveratrol was not successful in the clinical arena as an anti-cancer agent is that physiologic amounts of thyroid hormone in fact block serine 15 phosphorylation of p53 that's essential to resveratrol's actions. Here is some additional evidence that thyroid hormone is anti-apoptotic. Here we're looking at C6 glioma cells, rat glioma cells, and these are the nucleosome, as you know, as an index of apoptosis. And what you can see is that, do we have a, oh, here it is. What you can see here is that resveratrol is pro-apoptotic, induces nucleosome accumulation, and as soon as you add thyroid hormone, you block the resveratrol effect on apoptosis. If we look at nucleosome ELISAs in BHP27 and 1821 cells and FTC236 cells, these are thyroid cancer cells, where thyroid hormone is protecting thyroid cancer cells from apoptosis. You see a similar story where resveratrol induces apoptosis, and as soon as we add T4, we block that effect. Here we're looking again at apoptosis, and we're looking at some specific proteins here, BCLXS, for example, which is pro-apoptotic, and we can turn that on with resveratrol, and as soon as we add thyroid hormone, we lose the effect. So that it's not just serine 15 phosphorylation of P53, but there are other actions involved in the anti-apoptotic effects of thyroid hormone. Here we're looking at U87MG cells. This is a human glioblastoma cell line, and we're looking again at BCLX S, the short form, and it's pro-apoptotic. Its gene is a target of thyroid hormone. Tetrac is a deaminated analog of L-thyroxine I showed you, and it blocks binding of T4 to its receptors. So we'd introduce the concept in this particular slide that if we add T4 here, we don't see any pro-apoptotic activity here. If we add T4, we see some increase in BCLXS activity with T4, and with tetrac, in the absence of T4, we see we see a pro-apoptotic activity. So here is tetrac acting alone as a pro-apoptotic agent. So not only does it block T4 at the level of the cell surface receptor on integrin alpha V beta 3, but in fact it has effects, a life of its own in the absence of T4 and T3 on the BCLX system. Here is a cartoon of thyroid hormone and resveratrol competing on a cartoon of a cell. Resveratrol turns on MAP kinase, phospho-ERKs 1 and 2 result, and this phosphorylates serine 15 of P53, as I said, and leads to apoptosis. If we look at thyroid hormone acting at alpha V beta 3, what we see is a block of this phospho-ERKs 1 and 2 action on P53. If we look at resveratrol and thyroid hormone and the integrin, in fact, we find on X-ray crystallographic modeling that the resveratrol binding site and the thyroid hormone receptor site are right on top of one another here. So it's not surprising that thyroid hormone and resveratrol interact. They do not interact however, in the sense of blocking binding. They interact downstream of the receptors, as I said, at the level of Src kinase and MAP kinase. Now, thyroid hormone and T4 also inhibit FAS-induced apoptosis in mouse hepatocytes. And here are studies done by David Carpenter in Albany in a very elegant set of studies in 2006 showing that thyroid hormone, both T4 and T3, T3 in unphysiologic levels, T4 in physiologic levels, inhibited FAS-induced apoptosis. T4 also prevents caspase 8 induction or activation by the FAS TNF-alpha, a pathway that leads to apoptosis. And these are complicated slides, but the critical issues here are that in the presence of thyroid hormone, you lose the TNF effect and the FAS effect. They disappear. Now, EGFR, the epidermal growth factor receptor, EGF it turns on this receptor as it should, and you can see here it causes cell proliferation via the receptor. If we add T4, we in fact see increased EGF action. And this includes additional studies done by others. This includes anti-apoptosis. So thyroid hormone can act via FAS, it can act via P53, it can act via the EGF receptor, all to block apoptosis. And then the vimentin gene is a target of thyroid hormone, and you can see that the vimentin gene is turned on to a modest degree in these studies, and it rescues cancer cells from apoptosis. Finally, thyroid hormone blocks the pro-apoptotic actions of Velcade in myeloma cells. Now, Velcade is a reasonably effective anti-myeloma treatment, standard treatment, and in these studies done by our collaborators Karen Cohen and Osnat Asherfabian in Israel, and published recently, what you can see is that the apoptotic action of bortezomib, which is healthy in cells that are not seeing thyroid hormone, is in fact knocked down in the presence of physiologic quantities of T4. So I can summarize here by saying the points of action of L-thyroxine on apoptosis pathways are the following. On the extrinsic pathway, it affects FAS and actually affects FAS ligand binding. Here, it affects caspases. I haven't shown you the data for that, but they're published. On the intrinsic pathway, it has a variety of effects that involve p53 activation. And it also has a variety of effects on the mitochondrion itself, on a mitochondrial membrane, permeability, stabilizing the permeability of the mitochondrion, so that in fact the freeing up of cytochrome C and the release of cytochrome C does not occur. In summary, I can say thyroid hormone, particularly T4 at physiologic concentrations, is anti-apoptotic in tumor cells. Its anti-apoptotic activity is largely or exclusively expressed at integrin alpha V beta 3. The mechanisms of the anti-apoptotic actions of the hormone are multiple. They're incompletely understood as yet, but they involve both extrinsic and intrinsic pathways of apoptosis, and they affect expression of a large panel of genes that are relevant to apoptosis. Failure, and this is a paper that we just published in January, has conferred important clinical benefits on patients with advanced cancer. And this in part may reflect the removal of the anti-apoptotic action of L-thyroxine. What do I mean by euthyroid hypothyroxanemia? We give the patient methimazole to shut down hormonogenesis in the thyroid, and we give the patient T3 cytomel rather than T4 replacement. And so we create a setting where there is no endogenous T4, there is no exogenous T4. The patient is euthyroid on T3. And what we see is, in a group of patients now, 28 patients reported, what we see is stabilization or regression of tumor. We believe that this, in part, relates to the anti-apoptotic activity of T4 and its removal in this particular clinical setting. Questions?
Thank you very much. That was very exciting.
In regenerating tissues, the alpha V beta 3 is mainly expressed on angiogenic blood vessels, and I'm just wondering what types of, are the tumor cells themselves expressing alpha V beta 3, or is it the stromal cells, or is it the transform cells, or is it the blood vessels, and what is actually being targeted?
Yes, and yes. Anything that's rapidly dividing, and this includes normal cells at the site of an injury, for example, are going to express activated alpha V beta 3. That's the extended form of alpha V beta 3, and they're going to increase the numbers of molecules on the cell surface. There are about 7,000 to 8,000 alpha V beta 3s per cell, and that can go up to 35,000 when the cell is actively dividing. And not only that, its state changes. Rather than being folded over on itself, it extends itself into an active form, which exposes a host of binding sites for osteopontin, vimentin, what have you, that are important to the structure of the new tissue that's being formed. I've shown in other settings that the anti-angiogenesis piece is very strong of tetrac, the pro-angiogenic piece of thyroid hormone, is very strong. And so that we can essentially block that in the tumor cell with tetrac or with a nanoparticulate form of tetrac that we've put together. But it's a good question.
And sorry, second question, what were the type of tumors you were treating in your paper in 2015?
Oh, yes, they included glioblastoma, non-small cell, lung carcinoma, renal cell carcinoma, and sarcoma. So it was all comers. Another Israeli group has just put together a second contingent of patients, about 30 patients with euthyroid, hypothyroxanemia, with results that look on preliminary analysis similar. I'm sorry.
Is that mean the increasing of T4 is the indicated a bad prognosis with patient receiving with chemotherapy? Is it?
Did you get?
If the patient have a higher of T4 or especially increasing of T4, is indicated bad prognosis if the patient receiving chemotherapy?
They have hyperthyroid.
Yeah, hyperthyroid.
Yeah, it's clearly, I'm concerned about the upper half of the normal range of thyroid hormone levels, free T4 levels, as a risk factor for pushing the hormone ahead, pushing the tumor ahead in the patient. There are a series of reports of hyperthyroidism in cancer. There aren't a lot of them. There are a number of papers on hypothyroidism in cancer showing that tumor cells behave much less aggressively. This includes a huge series from MD Anderson in breast cancer. It includes 3 series from Europe dealing with hypothyroidism induced by tyrosine kinase inhibitors. And the patients who do best on TKIs when they have renal cell carcinoma are those who have the side effect of hypothyroidism. Cleveland Clinic has published glioblastoma data and also head and neck cancer data where hypothyroidism changes the course of the disease. So we don't have a lot of information about higher levels of thyroid hormone, but those data that are out there suggest that it supports the tumor, but the data are incomplete.
So we know that chemotherapy is effective in some settings, but I'm wondering if there's any evidence that thyroid hormone signaling is up in chemo residual as well.
Is up in the leftover cells? Yeah, we don't know, and it's a very important question. I like that. Yeah.
So resveratrol was originally hypothesized to activate SIRT1 and histone deacetylation as
Sure.
an anti-aging compound. So I know that since then it's been shown to be a little less specific than originally hypothesized, but I'm still curious, is there any connection between integrin signaling and SIRT1 or histone deacetylation?
Not to my knowledge. Yeah, it's a good question too. SIRT1 was a key issue for the purchase by Glaxo of the resveratrol story because it had to do with aging and carbohydrate metabolism.
Of course, I'm very intrigued by the multiple functions of integrins and working on osteopontin.
Yes.
The question comes to mind, how does the ligation of integrin alpha V beta 3 by thyroid hormone compare to its ligation by osteopontin? Osteopontin can be anti-apoptotic, so do they compete? Do they synergize? Do they antagonize each other? And as maybe a second question, we know that there's a difference in integrin signaling between an adherent and a de-adherent cell. Do we know whether it makes a difference, whether your thyroid cancer is in its original site or it's disseminating and in the bloodstream? Do we know if there's a difference in signaling by thyroid hormone?
We don't have enough information on the inside out or the outside out signaling of thyroid hormone affecting osteopontin, vitronectin binding, for example. We don't have information about that. We know a little bit about vimentin, and we know a fair amount about laminin. And thyroid hormone really improves the interaction of laminin and the integrin. Okay, so, and that's useful to the tumor. It's useful to normal tissues, of course. It's critical to normal tissues, but it's particularly important to the tumor. By the way, thyroid hormone also turns on matrix metalloproteinases. So even though it can stabilize a tumor, it can also set the stage for metastasis and for increased blood vessel formation by the MMPs. And so it indicates the complexity of thyroid hormone's potential contributions to the cancer. Now, was there another question?
But I think, yes, that answers the de-adhesion, yes. The
Yes.
de-adhesion part, that was
Yes.
part of the question.
Yep. It's a stunning hormone and a good hormone, wound healing and normal tissue growth and normal brain development. Everything it is so important there. And then it takes off its good costume and becomes bad in the setting of cancer. Okay.
Okay, well,
Good questions. Thank you very much.
thank you so much. Could I...